We're almost at the end of our exploration of human biological variation in regards to sex. We began with a discussion about genetic sex and how it is established at the time of fertilization. We then talked about gonadal sex and external genitalia. While our language divides babies into boys and girls, sex differentiation in all three of these dimensions is not so clear-cut. Sex differentiation is actually a complex trait, and like all other complex traits, there is variation. When we first talked about sexual dimorphism at the beginning of the semester, it seemed like such a simple thing, hey? Gorilla males are twice the size of females. Human males are 5 to 10% larger than females. We even talked about skeletal traits that are used in forensics and bioarchaeology to differentiate women and men. Men typically have more pronounced brows and chins. The pelves of women and men typically differ as well, with women having wider pelvic inlets and outlets that make childbirth more feasible. While this seems so straightforward, as you have likely come to realize, nothing is ever that simple in biology. Skeletal shape is not a dichotomous trait. It doesn't fall into either-or categories. A skull isn't clearly male or female. You can make an educated guess, but there is a lot of overlap. And pelves are the same. Let's look at some data. In a paper published in 2017, the authors explored sexual dimorphism in pelvic size and shape. For comparison, they first plotted body height on this histogram so that you could see how height varies in their sample. Males are plotted in blue and females in pink, of course. The overlap is shaded in a darker pink, kind of a purple, and you can see that as they come close to each other in these distributions. So you can see from this histogram that males are on average taller than women, but there is a lot of overlap. Then the authors presented pelvis size on a second histogram. You can see that here. There is a lot more overlap compared to what we saw with height. Notice how much of the graph is that dark pink kind of purpley shade, or pinkle? Anyway, indicating that shade indicates the overlap in pelvic sizes of men and women. Male variation is actually entirely subsumed by female size variation. And then there is pelvic shape. The shape of the pelvis has much less overlap between males and females, but notice there is still some overlap. Does this histogram, this bimodal histogram, does this remind you of a graph you've seen before in this module? Yeah, this one. What we see time and time again is that even in phenotypes that are the most differentiating between male and female, there is still overlap. Sex differentiation is such a complicated cascade of events with such a mix of genetic, hormonal, and environmental factors that differentiation is never absolute. We have talked about the differentiation in the early stages of development, fertilization, and during the first trimester of gestation. Now, let's look at sex differentiation in brain development. The previous video introduced you to the biological influence on gender identity. For this video, we'll focus on sexual orientation. A good place to start is with this guy, Alfred Kinsey. He was born in New Jersey in 1894 and studied insect behavior for his doctoral research at Harvard University. In 1920, he moved to Indiana University, where he taught biology. In 1938, after 18 years of teaching about insects, he was instructed to take over teaching the course on marriage. In those days, a course on marriage was really a course about sex. Professor Kinsey was immediately struck by how little scientists actually knew about the sexual lives of humans. He could teach his students a lot about insect sex, but he was at a loss to teach them about our own species. And so he set out to rectify that. Ultimately, he published 2 research tomes. In 1948, Sexual Behavior in the Human Male, and in 1953, Sexual Behavior in the Human Female. These 2 books were nothing short of revolutionary in the world of human biology. Never before had someone collected so much data about what people actually were doing in their sex lives. There was lots of talk about what they should be doing, but not about what was actually going on. Kinsey changed that. These books were based on over 18,000 interviews. His research assistants were highly trained. They asked each interviewee 350 questions. From the answers to these questions, Kinsey developed a scale of sexual orientation. The scale went from 0, being maximally heterosexual, to 6, being maximally homosexual. The 350 questions and the answers to them were all coded. You can see an example of the data collection sheet here. Kinsey and his team took incredible effort to ensure anonymity, and through that, the hope of getting honest answers. It might come as a surprise to you that people sometimes lie about their sex lives. Let's take a look at some of the results he found. The data showed that 63% of males and 77% of females were exclusively heterosexual. The data showed that 2% of males and 1% of females were exclusively homosexual. 35% of males and 22% of females were bisexual in their adult lives. Now, you might have had the perception that people back in the 1940s were pretty sexually conservative, but don't forget that these were the days of Marilyn Monroe, Sophia Loren smooching her sister Maria there, Mick Jagger on a date in Venice, and that's Kirk Douglas and 2 friends, and my great-aunt Margaret. Her stories attest that these exciting lives, that was not just for famous people. What about today? What are people doing today behind closed doors? Some scientists have argued that Kinsey's data were maybe a bit biased, that the people who were willing to answer 350 questions about their sex lives might be sampling out of the general population in a somewhat biased way. And you see that the bisexuality data may demonstrate such a bias. But what you also see is that a whole lot of people are not 0 on the Kinsey scale, maximally heterosexual. There's definitely variation in sexual orientation. More recent survey data in the United States show that 2.7% of men have had gay sex in the previous year. 7.1% of men had gay sex at some time in their adult lives. 1.3% of females had lesbian sex in the previous year, and 3.8% at some point in their adult life. So a question about all of this variation in sexual orientation. Is there evidence of a genetic contribution to sexual orientation? Perhaps people's willingness and interest in having sex with people of the same gender identity, perhaps that's cultural. Perhaps people's sexual orientation is influenced primarily by what they see around them. Or perhaps sexual attraction is influenced by biology, by genetics or not genetics. Does a person choose where they focus their sexual attraction, like the clothes you choose to put on every morning? Or is sexual orientation innate, like your height or your eye color? The answer to this clearly has a lot of social implications in today's world. And so, not surprisingly, there's been a lot of research to see if there is a genetic contribution to sexual attraction. The results come down quite resoundingly on the side of biology. For example, brothers of male homosexuals are 4 times as likely to be homosexual as well, compared to controls. Monozygotic twins, identical twins, are concordant for male homosexuality 52% of the time. Dizygotic twins, fraternal twins, only 22% of the time. Now remember, monozygotic twins have identical genome sequences, whereas dizygotic twins share only 50%, like other siblings. If genetic variation is strongly influencing a trait, the expectation is that monozygotic twins, identical twins, will be more phenotypically similar than dizygotic, fraternal twins, and in sexual orientation, that they are. Monozygotic twins are concordant for female homosexuality 48% of the time, 16% for dizygotic twins. So the same result is found for male and female homosexuality, though the levels of correlation are a little bit different. Now, a key point to keep in mind is that more than half of the monozygotic twins were discordant in spite of identical genes and similar rearing. Clearly, then, other factors contribute. Now that we've seen clear evidence of a genetic contribution, what does that genetic contribution look like? Is it due to one gene, several genes, or many? Is sexual orientation monogenic, oligogenic, polygenic, omnigenic? In 1993, Dean Hamer, a scientist at the National Institutes of Health, found that 28 gay men shared an allele on the X chromosome 75% of the time. Straight men had a different allele 75% of the time. This was pretty powerful evidence, and evidence that the linkage was on the X chromosome. Because it was on the X chromosome, that suggested that sexual orientation is maternally inherited. Now, a 1999 study did not replicate those results, but then a 2014 study of 409 pairs of brothers did. Well, in 2005, Mustansky and colleagues looked at the DNA sequence of 456 individuals from 146 families with 2 or more gay brothers. Now, they did not find a link with any alleles on the X chromosome, but they did find an association with a gene on chromosome 7 near the vasoactive intestinal peptide receptor type 2. Whew! A mouthful, huh? Or an earful for you. This gene functions as a neurotransmitter and neuroendocrine hormone. This gene is related to a hormone that is anti-Mullerian. So probably it turns on genes that masculinize the brain, or it's involved in that process. The results for all of these studies I just mentioned, they're conclusive in that there is a genetic influence, but they are not conclusive in terms of a specific gene. So perhaps it's because there's more than one. There's also evidence that sexual orientation is influenced by biology, but not genetics. In 1997, 2 biologists, Ray Blanchard and Anthony Bogart, found that sexual orientation correlates with the number of older brothers a man has. Each additional older brother increases the odds of being homosexual by about 33%. This led them to propose the idea of fraternal birth order influencing sexual orientation. The idea is that birth order reflects the progressive immunization of some mothers to Y-linked minor histocompatibility antigens by each succeeding male fetus, and the concomitantly increasing effects of HY antibodies on the sexual differentiation of the brain in each succeeding male fetus. So it involves mom's immune system and a hormone that only male babies produce, not female babies. In 2018, Blanchard and Bogart, they published the authors of the original paper from 1997. They were co-authors on a study that presented somewhat more direct evidence in support of the fraternal birth order effect. Let's take a look. The authors found that women's antibodies to NLG-N4Y increases with the number of sons she has gestated. Now, it's still a correlation, as the mechanism still isn't exactly understood, but in the schematic illustration that you can see here, you can get a sense of how this might be working. With the first son, the mom's immune system produces some anti-NLG-N4Y proteins. Her body does not do this for her female baby, but with her subsequent son, her immune system evokes a stronger response to that protein. And the next son, the next later-born son, her body evokes an even stronger immune response to NLG-N4Y. The accumulation of anti-NLG-N4Y antibodies would then increase the relative incidence of gay orientation among subsequent sons by inhibiting expression of the male-specific protein NLG-N4Y that is involved in neurological development. So mom's immune system is influencing the neurological development of her sons. Let's go back to the question about genetic influences. As with so many topics we have covered this semester, the advent of whole genome analyses, and especially the ability to analyze sequence data from many, many people, this has been a big deal in studies of sexual orientation as well. In August of 2019, a very large study was published that got a lot of press attention. Let's check out what they did and what they found. Have you sent your DNA to 23andMe, the company that will tell you all kinds of things about yourself based on their algorithmic analysis of your sequence variation? Well, if you have, amongst many other things, you may have answered a survey. So 23andMe asks participants to fill out a sexual orientation survey that includes questions like, how do you identify or think of yourself? To whom are you sexually attracted? With whom have you actually had sex? These questions formed part of the large study I'll tell you about. In common, these data were combined with data from a number of other large data sets that we call biobanks, where there's a lot of genetic information and a lot of different kinds of phenotypic information. So scientists, genomicists, can go in and probe those data in an exploratory type of way. The analysis relied very heavily on a biobank in the United Kingdom that's called the UK Biobank, perhaps not surprisingly. The researchers analyzed the genomic data of close to 410,000 people who had answered the question, have you ever had sexual intercourse with someone of the same sex? The researchers also included the genetic and phenotypic data for 75,000 people who had participated in the 23andMe program and answered questions about their sexuality. Now, the researchers had to dichotomize the 23andMe questions so that they matched what had been collected for the UK Biobank population. So it was that question, the answer to the question, was turned into a yes-no behavioral phenotype. The researchers also included about 15,000 people from 3 other data sets: the National Longitudinal Study of Adolescent to Adult Health, Molecular Genetic Study of Sexual Orientation, and the Child and Adolescent Twin Study in Sweden. In total, their analysis looked at the genomes and phenotypic data for about 500,000 people with the aim of identifying genomic loci that correlate with the answer to the question, have you ever had sexual intercourse with someone of the same sex? Please note that the study only included people of European ancestry, which is a pretty limited subset of human genetic variation, as you know. Keep this in mind. It is a lot of people, but it is not a good sample of human genetic variation. Similar to the results from earlier studies, the researchers found that lots of people are heterosexual, but that a significant number of people are also engaging in sexual activity along a spectrum from homosexual to heterosexual. They also found some trends in the phenotypic data, the behavioral data. First, let's look at this graph here. Year of birth is indicated on the x-axis, and percent of individuals reporting same-sex sexual behavior is on the y-axis. For many men and women, the percentage increases over time, indicating that more people born in later years report having had a same-sex interaction. For women, this went from about 1% for those born in 1940 to about 6% for those born in 1970. For men, it was just over 2% of those born in 1940 and close to 8% for those born in 1970. The researchers also found genomic loci that are associated with variation in these behaviors. The graph on the left is a Manhattan plot that you are familiar with from earlier in the semester. The x-axis indicates the chromosomal position, and the y-axis indicates the statistical likelihood that genetic variation at that loci correlates with phenotypic variation. Let me make this bigger for you so it's easier to see. When a dot is above the dashed line, it is considered significantly associated. The interpretation is then that chromosomal region contains a gene or regulatory element that influences the phenotype you are looking at. This graph shows us that genetic variation at 5 loci is associated with the response to the question, have you ever had sexual intercourse with someone of the same sex? Of these 5 loci, 2 are significant for women and men, indicated with the red diamond shape. 2 are male only, indicated by the downturned blue triangle, and 1 is female only, indicated by the dark blue upturned triangle. Is this, then, the answer to our question about how many genes influence variation in sexual orientation? Is it 5? 3 or 4, actually, depending on whether or not you're male or female? So does this mean that sexual orientation is oligogenic? Well, these data definitely do show that genomic loci influence sexual behavior. But, of course, it isn't quite so simple. Remember, the phenotype being analyzed was the answer to, have you ever had sexual intercourse with someone of the same sex? There were a bunch of other behavioral data analyzed in this study as well. This graph is a little hard to see because there's so much packed into it, so let me walk you through it. And to facilitate that, I'm going to spin it clockwise so that we can make it bigger. The x-axis is now on the left rather than at the bottom. And the x-axis indicates the genetic correlation between a range of different traits and the answer to the question, have you ever had sexual intercourse with someone of the same sex? There are physical traits on the listed across the top. So physical traits over on the So there are phenotypic traits at the top, physical traits on the left, or then reproductive traits, personality traits, mental health, and risky behaviors. Below each trait listed is a dot with a line through it, going from top to bottom. The dot indicates the estimated genetic correlation for females in blue and males in that turquoise color. So females are always on the left and males are on the right of the pairings. The dot indicates the estimated genetic correlation, and the line indicates the margin of error. If that line overlaps with 0, which is indicated by the black dashed line, then there is no significant genetic correlation between that phenotype at the top and the answer to the question, have you ever had sexual intercourse with someone of the same sex? Now, I keep saying that the phenotype is the answer to that question because the phenotype isn't actually sexual orientation. Because what the researchers have done by using that question is maybe not actually gotten an indication of someone's sexual orientation, but perhaps something else. Because is it possible to have ever had sexual intercourse with someone of the same sex, but not actually identify as homosexual? Let's look at these genetic correlations. Notice that the dots that are farthest from the dashed line from a genetic correlation of 0 are number of sex partners for men and women, openness to experiences, loneliness, cannabis use, smoking, quote-unquote risky behavior. I'm not actually sure what they mean by that, but we can probably guess. And also genetically correlated with some other personality and mental health traits. So, I ask you, are these genomic loci specifically influencing sexual orientation, or are they showing us that at least whether or not a person has had a sexual experience with someone of the same sex at least once in their life is a complex trait intertwined with personality? Another way to think about sexual orientation is from a comparative perspective. Is homosexuality, is bisexuality, are these behaviors unique to humans? The answer is no, not at all. You can find examples all around. You already know about bonobos. This is a sister species to the chimpanzee in the genus Pan. Bonobos use sex as a way to ameliorate social tensions. Male-male, female-female, young, old, all the time. There are pairings of sexual interactions that demonstrate a wide range of variation in sexual orientation and attraction. The wider range of sexual orientation in bonobos, this is an evolutionary adaptation for resolving social stress and for increasing social cohesion. You might be less familiar with the Malaysian albatross. Field studies have shown that 31% of the pair bonds on the island of Oahu are two females. These two females do all the same things as male-female pairs, but both females may step out of their bond to be fertilized, and then they come right back. Scientists think this may exist because there is a bias in the sex ratio for these albatrosses. There are more females in the population than there are males, and consequently, this is interpreted to be an evolutionary adaptation. There are also stories about penguins and other critters, and each of these examples tells us that variation in sexual orientation is common across the animal kingdom. And as we see with the bonobos and the Malaysian albatross, that variation offers an option for solving some dilemma. So remember, variation is the most fundamental part of evolution. Without variation, evolution and adaptation cannot occur. Since sexual orientation is a complex trait, there are options, evolutionarily speaking. In closing for this module, let me give you some more demographics. 0.6% of adults in the US identify as transgender. That's about 1.4 million people. 3.5% of adults in the US identify as LGB, lesbian, gay, or bisexual. That's about 9 million people. That is the equivalent to the population of the entire state of New Jersey. Now, within that 3.5%, about 1.8% identify as bisexual, and about 1.7% identify as lesbian or gay. 8.2% of adults in the US report having engaged in same-sex sexual behavior. And 11% of adults in the US report having at least some same-sex sexual attraction. So what to make of all of this? Well, sex is not a discrete trait. Well, neither is gender or sexual orientation, for that matter. Our language implies a dichotomy between male and female, but the biological distinction, it's not discrete. The boundary between male and female is a gray zone with lots of intermediates. And some languages and cultures incorporate this biological variation easily, and others of them do not.